What's good, what's good? Welcome back to another video. We're jumping into the kind of week 2 material, kind of cell structure. I would like to say, I think I messed up and I recorded the cell junction stuff under week 1 material. I think it's technically part of week 2, but that's okay. You can just watch them out of order. So, cell structure. This is going to be diving into all of the different components of the cell. It's a lot of just straight definitions. This does this. This is related to that kind of material. So let's go ahead and jump right in. We're going to be looking at the plasma membrane, the nucleus and its components, rough and smooth endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, ribosomes, mitochondria, cytoskeleton, all that kind of fun stuff. So all of this stuff that we see, all these different components of cells, as well as different kinds of cells. So in general, you have what's called, like, quote unquote, your typical cell. The typical cell contains some very specific things. Then we also have what are called specialized cells. So if we look at other cells, like erythrocytes, for example, this is one that usually pops up on test questions. Erythrocytes are a little bit different, red blood cells, erythrocytes, because they don't have any of the organelles. So that's a fun fact. People like to throw that in there. Nerve cells act a little bit differently because once we kind of produce nerve cells, our body doesn't continue to produce more of them. Skeletal muscle and smooth muscle do different things. So there's lots of different, I would say, irregularities to cellular structure. But we're going to look at the general, aka the quote-unquote typical cell. So generally, all cells have common structures and functions. Specifically, we're going to look at the plasma membrane, cytoplasm, as well as the nucleus. So the plasma membrane, we've mentioned it a couple of times. It's that outermost layer. It's what surrounds the whole thing. It is what is known as a semi-permeable membrane, which basically just means that it lets certain things in, doesn't let other things through. It controls what goes in and what goes out, which we want it to, because otherwise it'd be leaking stuff all over the place, and that would not be good. Cytoplasm. We talked about this before as well. We'll mention this a couple more times. Our body's mostly water, and we need, water and stuff, or something to keep us all held together. Because if we were just straight water, obviously we just turn into a little puddle with a bunch of clumps of cells. But we're not that. So cytoplasm is kind of like the jelly-like substance. It's an intracellular fluid which contains the organelles and all the other stuff. And then, of course, we have our nucleus, which is that control center. So this is kind of the general stuff. We'll cover most of these. If you want to take a moment, again, look at the description below if you do not already have my notes. Otherwise, make sure that you go and you download the latest version of everything. I am slowly updating these as I come out with these videos. So if you download the notes that you have purchased and I've sent out, and you're not seeing all of this information, make sure that you go and check and download the most updated version. This is just straight definitions of all of this stuff. This is perfect. Copy and paste that into Quizlet. Make a little Quizlet so you can review just the straight definitions and recognize some of the keywords, of each of these organelles, just so that you know in general what they are and kind of what their function is. This was a lot of the test questions are going to come from, is what is it, what is its function, and then if there's anything like special specifically about it. For example, the plasma membrane. There's lots of stuff we can talk about with the plasma membrane. Most importantly, again, we want to know what it is and what its purpose is. So the plasma membrane, most often we're always going to see it's called the phospholipid bilayer. We talked about this already in last week's stuff, or in week 1, the first chunk of material, where we have a phosphorus head, which is hydrophilic, likes water, and then we have a hydrophobic tail, being a fatty chain. So phospholipid. and there's two of them, so it's the bilayer. So this is, I said that keyword before, semi-permeable, which means that it controls what goes in and out. Within all of the plasma membrane, right, we have a bunch of different components and other stuff mixed in with it to make sure that it is stable, that it can do its job, and also that it can let stuff go in and out. There are a lot of different things. There are certain things, that can just directly cross through the barrier without any help, but other things need assistance. They need what are called intramembranous proteins. So we can see some of these in here. We have some stuff. We have glycoproteins, we have glycolipids, we have cholesterol, we have integral proteins, all of these kind of things. Down below, again, I threw in here a bunch of those definitions. Biggest thing to remember is that phospholipid bilayer, what it's made of, what part is hydrophilic, what part is hydrophobic. That's the big key takeaway for plasma membrane. Again, there's a lot of information on here, and I kind of try to explain these through. Biggest things you want to remember are what are here bolded, and then most of the rest of this is just kind of explanation of these specific points. So it's a dynamic barrier between internal and external structures, internal and external environment. Dynamic meaning that it lets stuff go in and out. Stuff is constantly going in and out of our cell membrane. Nutrients, waste, signals, all sorts of stuff in order to keep everything at homeostasis or balanced, as well as functioning appropriately and making sure that it's transmitting the signals and stuff that it needs to transmit. It's also big job is to maintain chemical and electrical gradients. Essentially, this is talking about concentrations. We need a specific chemical concentrations of different things in and out of the cell at all times. So through the plasma membrane is how we manage that. As well as electrical gradient, we need specific charges, electrical charges or voltage on either side of the cells in order to make sure that everything is working appropriately. Part of this, think, inside of the cell is typically negative. It's overall negative because most of our amino acids, our proteins, tend to be negative, and we have a lot more proteins inside of our cells than we do have just outside hanging out. Only thing that's additional on this slide here is talking about this fluid mosaic membrane. So a fluid mosaic model, it's just describing the membrane as a flexible dynamic structure where you have all those different components that are able to move around within the bilayer, similar to how tiles in a mosaic shift. So it just allows for the membrane to constantly be adapting to the different conditions, different environments that it's in. Whenever something changes, in order for allow it to adapt and continue to survive. Our bodies are all about adaptation. Remember, we have this word again, amphipathic. Amphipathic means that it has both, of those hydrophilic and hydrophobic properties. I rewrote it down here towards the bottom as well as going through kind of what the integral, there we go, if I can talk, integral proteins are. So those are proteins, they're the ones that are responsible for functions like transporting receptors, doing cell signaling, that kind of stuff, which we'll see in the next slide. So we have integral proteins, we have extrinsic, aka peripheral proteins, and we also have glycoproteins. So the integral proteins are our transmembrane proteins. They help transport stuff. Boop. The extrinsic or peripheral proteins associated with either cytoplasmic or extracellular region of the membrane. These are proteins that they're going to be technically part of the plasma membrane, but they're typically associated more either with the outside of the cell or the inside of the cell. We don't go into a lot of specifics about what they are, just understand, hey, there are other proteins that are going to do other jobs. In the future, we're going to talk about certain receptors and other proteins and stuff that are going to be these peripheral proteins, but we don't need to worry too much about those right now. We also have what are called glycoproteins. glycoproteins right in there, which these are sugars which act as a recognition site. So now remember, we have receptors. A lot of our signaling, our cell signaling, is done through chemical receptors. So chemical will come and it binds to a protein that has a receptor site in order to cause some type of reaction to occur. So this is one of those examples. It has a recognition site. That's the job of the glycoproteins. It's also important for self-determinant for immune system. Essentially what that's saying is our body has specific glycoproteins, and if there are other cells or other stuff that enter our body, that come into our body, that have different glycoproteins that our body doesn't recognize, the immune system can then tag that and say, hey, that's not supposed to be here, and it can go ahead and eliminate it as a threat. Functions. This is lots of stuff kind of test-wise. You want to know the functions of stuff. So functions of the membrane proteins. Here's just kind of the whole list. Don't want that one. There it goes. We have transport, enzymatic activity, receptors for signal transduction, intercellular joining, cell-to-cell recognition, as well as attachment to the cytoskeleton and extracellular matrix. Now, if you stop and you think about it, we've already talked or mentioned most of these in previous lectures already, or as well, you're going to be getting into a lot more of this into biochemistry. So there's a lot of overlap between cells and tissues and biochemistry because a lot of what's happening histologically is related to the biochemical type reactions. So I went ahead and I kind of sorted out each of these and added a little bit of extra information, just kind of explaining what's actually going on. You do not need to go this deep with it, okay? Just understanding, these are the functions. That's really what you're going to be tested and quizzed on. Just if you want to understand it a little bit better in case, you might get a question that's a little bit deeper level, which, if you read through this and look at it, it probably will help you answer any questions that you'd get like that. Okay, this is now kind of the pause point. We're moving on to the second portion of this lecture, which is the organelles of eukaryotes. Organelles is just a fancy way of saying little organs. So think about like our body as a whole. We have our major organs, our heart, our lungs, our kidneys, our intestines, all that kind of stuff, each playing a specific role in our survival. Same thing now with these organelles. The organelles are the little organs of the eukaryotes, eukaryotes being multicellular beings. Each of these is going to have an important job when it comes to keeping us alive and functioning. It's not only just at the individual cellular level, but as a being as a whole. So, starting from the top, nucleus. So nucleus, its job is, it's kind of the control center of the entire cell. It regulates the cell because it contains the genetic materials, that DNA, which codes for our life and for all of our proteins and everything else that we need to have to survive. Within the nucleus, we also produce ribosomes. Okay, so we'll talk more about ribosomes later. The short story is that ribosomes help build proteins, and we need proteins for survival. As well as those ribosomes are split up into a couple of parts. So you make the parts separate, then they come together to make a full ribosome. Again, we'll look at that again in a little bit. But that happens, the making of the ribosomes happens inside of the nucleus. Here we have the nucleus, all that genetic information. Let me see. I think it talks about, I'll talk about it here. I don't think it mentions it later on. The nucleus is typically looks like a bigger circle within the cells. As you can see here, this whole big circle is the cell. We also have what's called the nucleolus. This is a key thing to differentiate because it's going to be asked on test questions, especially like the lab portion, where with the lab portion, you're having to recognize it's going to show arrows. So it might show this picture right here, and it puts an arrow like what's the whole thing, or like what's this specific structure in here. Whole thing is going to be the nucleus, the whole big circle, and then the more dense part inside of the nucleus is known as the nucleolus. Okay, so just differentiating those words, try and keep those separate. Additionally, some other components, I'm actually going to switch these so we can go over all the terms before looking at the pictures. We have a nuclear envelope. This is basically, so just like a cell as a whole, you can kind of think of the nucleus as like a cell within a cell. The cell as a whole has that double phospholipid membrane. The nuclear envelope, which just surrounds the specific nucleus, also has a double membrane. So it has its own membrane. Doesn't really show it here. I thought I had a picture where it showed it. We'll see if it comes up later. Biggest difference between this membrane and the cellular membrane is that the nuclear membrane, the nuclear envelope, has nuclear pores. It has a bunch of little holes all throughout it. And those holes allow the materials to transfer. Remember we talked about like those ribosomes and stuff, to go between the nucleus and into the cytoplasm to the rest of the cell. So that way, like, there's a little bit of that open communication between the nucleus and the cytoplasm and the rest of the cell. DNA, we talked about DNA a little bit before already. So remember, DNA is a double helix structure. 2 complementary strands of DNA. That's all of that at the golf course. A pairs with T, G pairs with C for our base pairs, and DNA is built like a ladder, and stabilized by the hydrogen bonding between those bases. Remember, the A and T have 2 hydrogen bonds, G and C have 3 hydrogen bonds. That's how they all stay together. We also get into just a little bit more kind of vocab type stuff. I have more here down below about like definitions and stuff, but basically it goes into, it just depends on what state the DNA is in, is how we identify it. So, for example, DNA, if it's just all loose, one single strand, it's DNA. If we start to package it, so start to fold it up a little bit using these special proteins called histones, which we'll get more into later, if it's folded with some of those histones, we call it chromatin. Now there's 2 kind of subcategories of chromatin. If it's super tightly coiled, densely all wrapped up with these histones, we call it heterochromatin. If it's a little bit looser, a little bit more open, we call it euchromatin. And then finally, if like the whole chunk of DNA, all of it is packed really, really tightly into its super condensed form, and we can clearly visibly see it, we call those chromosomes, which you've probably heard the terminology chromosomes before. And again, we'll actually get into more about chromosomes, kind of what they are, what they look like, in the week 3 material, lecture 3 type material. Okay. So I'm actually going to draw, let me do this. I'm going to insert a new slide right here. Let's do a blank one. So we have DNA. Once it gets folded, chromatin, so DNA. DNA goes to chromatin. Chromatin goes to 2 separate ones. We have heterochromatin. and we have euchromatin. And let's add in histones. Jeez. I need one of those little pen pads. Histones, add in histones. I think there's an E. Histones. Here we go. DNA plus histones gives us chromatin. And then chromatin DNA, when it's fully packed all super, super tightly, they're chromosomes. So, just kind of the flow of it. DNA is a single strand. Chromatin is once it's been packed with histones. And we can call it either heterochromatin or euchromatin, depending on how active it is. And when we super package it all, prepping for cell division, we call them chromosomes. There's our nucleus, nucleolus, nuclear envelope, and you can see the nuclear pores. They wouldn't have to identify this stuff. Here you can see the nuclear pores, all those little dots. Now here's actually getting into more of that chromatin packing. So there's that histone, is a type of protein found in the chromosomes. It binds to DNA and gives chromosomes their shape and controlled activity of genes. Talking about like a little bit deeper, and here's more of this. The DNA gets linked with the chromosome, linked with the histone, and it kind of becomes, you get this small structure called a nucleosome. I think I should have definitions all in here somewhere. I guess I didn't put those. I'll put them down in here and then re-upload this. So chromatin packing, so you have DNA, add in histones. Specifically, we get 8 of them equals a nucleosome. Add several nucleosomes together. That gives us a chromatid. And then the chromatid, keep folding it all up, and that gives us the chromosome. Just again, a lot of vocab. Most of this, what we really want to focus on, is understanding the names, link the names to chromatin, to chromosome type, to packing it, okay? To the formation of the chromosomes. We want to know, it's from DNA, add in histones, it keeps on packing. If we see the word nucleosome or chromatid, we can think in order. DNA's first, then we get added in the histones, and then it becomes a nucleosome. A little bit bigger, it's a chromatid. Even bigger, we can call it a chromosome, kind of working, scaling up in that way. And this shows us, we talked about how, certain proteins have tertiary or quaternary structure. Each individual histone is complete at tertiary structure, but then they all come together in a quaternary structure with 8 of them together to all bind along DNA and to wrap it. So there we go. Nucleosome is the structural unit of DNA, which has a core of 8 histones. Let me keep on going. So just these little details that, that core of 8 histones within a nucleosome, what a histone is, and like the levels of folding is what's going to be testable material. Heterochromatin and euchromatin. So we can see the difference here. See how the euchromatin is kind of much more open, much lighter in color because it's not as dense. Whereas heterochromatin are these denser components because they are less active. There's the nucleolus. The nucleolus is the specific site of synthesis and organization of ribosomes. So making of ribosomes happens within the nucleus. Remember, the nucleolus is even smaller within there. So if we were to get a question that's something like, where are ribosomes synthesized? and you had two answer choices, let's say you had nucleus and you had nucleolus, a lot of times this is how teachers will get you, is they'll give you both of these choices, and you're like, oh, well, it said the nucleus. Again, nucleus is correct an answer, but nucleolus is more correct. So that's the one that we're going to want to pick because it's more specific. You won't get a ton of questions like this. You shouldn't. Some professors you'll get more than others, but just keep an eye out for that. I'm not going to spend a lot of time going through all of this just because there's a lot to all of this. I wrote out like a little bit of a description of each of these. Biggest things that you're going to want to remember is just the link of these. This is all you really need to know, this little component. You have these different regions, different parts of the nucleolus and different proteins where you have nuclear organizer, aka NO, which encodes rRNA. rRNA stands for ribosomal RNA, which is what kind of makes up the ribosomes. You have these things called pars fibrosa and pars granulosa. So this is what's written right here on the slide is what you really need to know. Down below is more of a description. Here's just now a picture of that, each of these individual components. Okay. Switching gears now, again, a little bit. I'm going to do it on time. I'm going to go ahead, I'm going to end the first video right here, ending at the nucleolus. And we will continue in part 2, getting into the endoplasmic reticulum components. So thank you so much for watching. Good luck studying, and I will see you all later.